So what is required for the similarity search? We have got similarity at the two levels. So we have already seen yesterday, similarity at the chemical level and similarity at pharmacophore level. So in the chemical level, the chemistry and the size and the shape of the molecule is having a similarity at the chemical level. But in the pharmacophore level, the molecules will be appearing as a different with respect to chemistry, but their pharmacological action is same. That is actually what we call it as a pharmacophore similarity. So what is required now for the similarity search? Both we have to do. That is the reason we need to do both do. But the way which we are going, the descriptors, whether it is a chemistry or biological, depending upon that, we need to use a software. So both are required. We need to use a different softwares for the chemical similarity and pharmacophore similarity. So a database SQL or NoSQL. So that is based on that Postgres, MySQL, MongoDB. So these are all the flat file descriptor for the chemical similarity functions. You can use the ChemFP. So chemical cartridge to generate the fingerprints, descriptors for the molecules we have got in the RDKit and OpenBabel. We can generate the descriptors with these software. Similarity function to calculate the similarity, we have got software like CADD, DICE, Persky. So this can be written in the multiple frames. So that is in the Python also, C++ and C. So inside the SQL database. In those cases, what we can do, we can apply the multiple structures at the different descriptor levels with the file types. So the file type may be, we have already seen in key types, which we are going to do, or we are going to have a mole files, depending upon what type of similarity we are going to see. For the 2D fingerprints, most of the times the molecules are represented as the binary vectors. Each binary vector will be having with 0 and 1. So each bit or the bit string, that is the binary vector, represents a one molecular fragment. So typically this length will go to 1000 bits and a complex moiety will have further. So the bit string for the molecule recorded as a presence of the particular functional group and absent of that particular fragment in another molecule B. So we are going to compare the same manner. Whether it is present, we assign the 1. If it is absent, it is 0. So this is what we have done in the Tanimoto score, which we have already done. See, the same thing is applicable here also. For the 2D fingerprints, we are repeating. So originally developed for the speeding of this substructure stretch. It is very faster in nature, but at the size and the volume level, we may have a difference. But the structural level, it is same. Chemical structural level, they will be same by this particular concept. So for the query substructure to be present in the database, molecule can set to a 1 in the query, must be set to 1 in the database structure, where we are going to have when both the fragments are present in the tested moiety already, which is present in the database, that is working as a drug-like moiety. So both comparison we are going to do. All the functional groups matching, we are assigning the number 1. Similarity is based on determining the number of bits that are common to two structures we are going to assign. So the molecular similarity, which we can assign by the two things, that is parameters, that is descriptor. One is a Euclidean distance, another one is a Tanimoto score or Tanimoto index. So it is also given in the Jacquard scale. So in these sequences are vectors, numeric values that can be compared with the similar distance matrix and similarity matrix. In the Tanimoto score, we assign the 1 and 2 by the molecule X and Y. And we see what are the common in nature. And we are going to calculate the Tanimoto score. The score which is above 0.85 is considered as identical. And below the 0.5 to 0.85. So it is not exactly identical, but it is less than 0.5 approaching to 0 means they are not identical at all. So there is no similarity between them. So to get the Torimoto score passed between the two chemical structures, we know that we need to have a score greater than 0.85. There is another parameter we can do in the chemical similarity. Molecular similarity is a Euclidean distance. Euclidean distance is the short path which is present between the two atoms of the molecule. So your molecule is containing multiple atoms. They are aligned in a different manner in the three-dimensional space. Say, for example, you are having a homologous series of your structure. When you see one end of your structure will be present, another end, another functional R group is present. The actual distance, if you see, is very large. But when the moiety is present in the three-dimensional array, they will have a short path distance. We need to consider for that to see the similarity. So the Euclidean distance is only taken as a axis and the edges for the calculation of the similarity in software. So this one we are going to see what is the difference between the two atoms, which we need to consider the short distance. We need to consider for the calculation of the Euclidean distance. If these distances are matching, it means we can say that the drug and the chemical we are comparing, both are same. There is another approach we need to go further, is 3D based similarity, where we are going to consider the shape as a parameter to see the similarity. One concept is called as the ROCS, rapid overlay of the chemical structure. So there is silicosit.com is present. It will shape the structure and find the similarity. So, but the 3D similarity is a little bit complex one and more expensive than the 2D similarity method. What we have got at Tanimoto and Euclidean distances, they are cheaper methods. 3D based similarity requires consideration of the conformational flexibility. So we need to see the rotational of that particular ligand, and we see the conformations flexibility. Most of the times it is having multiple conformations most of the times. So they will be behaving in multiple conformations. So that is the reason in certain angles only they will have a similarity. In remaining angles they will not find any similarity. So this is what the complexity in the 3D based similarity. So in the 3D based similarity, we can have two types of searches. One is a rigid search. So it is based on the single conformer. This rigid search is used in the key and lock type of thing, where we are seeing in the initial virtual screening and initial high throughput screening. We don't want to take the conformational analysis because it will be time consuming. First, we can assign the lead, and after the lead based on the key and lock system, not as a ball and socket like a flexible search. So the rigid search, it is a simple, and both the receptor and the ligand are the rigid in nature. So there is no conformational outlook here. That is the reason it is fast. So the another one is the nearness to the reality is the flexible search. So the conformation explored at the search time, where we are going to rotate at the multiple angles, and these angles which is having a different conformations based on the number of rotations you are going, what is the angle you are rotating it. So that makes the time, and assemble the conformers generated prior to the search time with each conformer and each molecule considered to run. So we need to see that how many conformations we are want to require. Based on that, we will see the how much degree you want to rotate, so that we will have a complete data without missing. So we can have the further extension of the learning in the YouTube. You will be finding so many videos in this. You can find the picture later. Multiple actives known, pharmacophore searching. Most of the times in the pharmacophore searching, not like a chemical search. In the pharmacophore search, the same moiety will have a functionality at the different receptors. So that is the one which we are going to explore the new area. So an assemble of the steric and the electronic features that are necessary to ensure the optimal supramolecular interactions with the specific biological target. we need to trigger or the block the biological response. We need to have this pharmacophore searching, where the pharmacophore is chemically dissimilar, but it is pharmacologically same, biologically same. So this is what we are going to have a different shapes assigned. Hydrogen bond acceptor, hydrogen bond donors, and aromatic rings, and acyl rings, and cyclic rings. So these are all contributing to some space. So in the drug design, the term pharmacophore refers to the set of the features that are common to series of the active molecules. So these series or active molecules are chemically different. So these hydrogen bond donors and acceptor positively, negatively charged groups, hydrophobic regions are typical features. They will contribute to the pharmacophore mapping. So these concepts are very much important. So in the 3D pharmacophore, so the 3D-dimensional pharmacophore specifies the spatial relationship between the groups. So we have got a multiple triangles where we are going to find the distance between the Euclidean distance between the two atoms in the question. And we see them for the comparison in the form of triangles. And the matching triangles in this picture, you see different structures are present. And most of the times, if you see here, when the first and the second structure from the left side, if you see that, you got pyramid in ring structures, and you got a pyridine ring structure in the two structures which are present. The Euclidean distances are both are same. The edges and axis for these two are appearing like a same, even though the molecules are particularly different. So the OON, when you see the distances, it is appearing to be the same in the OON, that is oxygen, oxygen, and nitrogen in the second molecule. So if you see the same, which is in the top of your slide, one picture is present with alkyl chain to the phenyl ring system. So where also you have got a OON. So the Euclidean distance, if you see them, it is same, even though the chemical nature of the molecule is different. But whereas the molecule, if you see here, where with the phosphate groups, this is obviously completely different. You have got a phosphate linkages to the systems. So the OOO distances are completely different. What we are seeing in the phosphate oxygens, where we are seeing, they are completely different. So these triangles, when it is being done, the machine learning system, it will compare the triangle edges, the distances, to see the similarity in the 3D pharmacophore matching. Now, what is the workflow we need to do? That is very important to us, how to proceed in the pharmacophore modeling. Collect a set of experimental compounds for the target. So we have got, say, a number of compounds. We know the target structure. Select the threshold for the activity values and mark compounds as actives and inactives. Create the training and test set. Depending upon the scores which are already present and available data, we are going to say it is an active and inactive. This data will be used for the machine learning training. So generate the confirmation, create the pharmacophore model. So where the actives are present, and it will give the idea that what of the conformations in the ligand that will be going to fit into the receptor, we know it. Similarly, the inactive will give the training model for the shapes which cannot fit into that. So this will get a good training, and the distances between the edges of a different molecules, it will already get trained in what distances they will be similar. The edges, they are similar. It will be knowing it. So they identify the essential for the activity of a molecule. What are the triangles where it is very important keen to be present in all type of the drugs in that particular protein. So we have to see that. So that is, we are going to fix for the further pharmacophore similarity search. So perform the statistical analysis of the model for the refinement, so that what is the prediction ability for the what we are going to gain with that particular interaction, seeing between the drug and the so-called ligand and the receptor. The same ligand is compared with the another chemical we are in the test, we are going to see. So ultimately what we are going to do here, there are specific features which are present in the multiple chemicals which are present as a pharmacophore. So the different chemicals are resulting in a similar activity. So the now parallelly we have to do the virtual molecules which are present, not which is already existing. That also we can try with a conformer database. This is actually what we can do with a fragment based drug design, what we have learnt. Similarly, we are arriving here also, and we can take the chemical databases also. And we have seen in the yesterday class, we need to do the paints files, we need to do the interfering molecules like paints, and we need to do the rare earth chemicals not possible to get in the reality, such thing we need to remove, and the salt forms we need to weed out and filter out. So any toxic chemicals which will give the false positive that we have to remove. So these are all the things which are going to give a list of moiety in the virtual space. We will conformer generation, we will make it, and 3D search we have to take, and it will act as a hit. Further hit will be modified by the SAR, QSAR to result into a drug. So we are going to have a multiple factors to see this, not only one. Sometimes it will be having these triangular spaces. So another time we are having a number of rotatable bonds and polar surface area also in the pharmacophore modeling. So what are the tools available in the pharmacophore searching if you want to find the similarity in the 3D level? Are one is catalyst, Accelris one, another one is a phage, that is a software by this Schrodinger, ligand, that is ligand scout, pharmagist, farmer, shafts. These are all the tools perform the pharmacophore searching, and they will find the similarity and assign the scores. So many actives and inactives are known in the machine learning methods. We are going to develop a structure activity relationship modeling so that the computer will get trained with known actives and known inactive compounds. So it will get trained based on what functional group it is creating in activity, what presents making it active. So this quantitative structure activity relationships, it will generate many hits further. So, and we can refine the molecular structure also by having a quantitative relationship where we have got a mathematical equations for each functional group changes with respect to your biological response. So the long established concepts what we have learned for long back is the Hans analysis and free initial analysis, where we are going to take these one for the similarity search. Generally restricted to the small and homogeneous test for the lead optimization, but we cannot use this particular Hans analysis and free initial analysis for the bigger moieties like proteins. They are the natural ligands which are present in our body. So the structure activity relationships can be drawn, where we are going to assign that by having this moiety, whether the activity is present or not, yes or no, qualitatively. So the data is usually qualitatively, it can be used with the data consisting of a diverse structural classes and multiple binding modes. Most of the times, some resistant to the noisy data is present in the high throughput screening. That is the reason we have got a filtering mechanisms and wash out functional groups. We need to reduce the number in the HT screen data. Resulting model used to update the compound for the lead finding, not to identify the drug candidates or the drugs, not like a QSAR. In the SAR, we are going to find out the lead finding. So we cannot find the drugs exactly, but in the QSAR, we can bring the drug with a multiple modification, which are going to work like a drug-like moiety. Ultimately, what we have arrived in that particular journey of a Wittebell screen, what we need to do the protein ligand docking. So this is what the picture is saying, the three-dimensional representation where we have got the ligand is fitting into the pocket. So the computational method, which mimics the binding of a ligand in the protein, we will see in the in silico model visually, and without going like in the high throughput screening, we are not going to use a natural proteins for the test in the laboratory. We are going to see in the computer lab itself, not in the bunch top. So it predicts the pose of the molecule, it binding in the site, and what is the pose, it will having a better affinity. And by affinity or the score is representing the strength of the binding. So when the poses changing it, it is resulting in a dissociation, then it will not be acting sometimes, and it will be acting as a antagonist sometimes, depending upon the dissociation association constants. So the pose and binding site. So the binding site is a active site which are present in the protein where particular amino acid is exposed for the interaction with the ligand. So the part of the protein where the ligand binds, that is called as active site. Generally, it is a cavity or a protein surface, can be identified by the looking at the crystal structure of the protein bound with known inhibitor or a ligand. So we have already told that we can find it by the X-ray diffraction tools and NMR. So these tools it will give after the cryo drying, so freeze drying. So we are going to get the interaction sites. And after getting the interaction site in the protein pocket, we need to find the better pose, that is binding mode. So the geometry of the ligand in the binding site, so the geometry, location, orientation, and the conformation of the molecule, that is resulting into the interaction, that is protein ligand complex, we have to see. So the protein ligand complex, how does the ligand, the small molecule bound into the active site of the protein? We have to represent by the docking. So docking algorithms are based on the two components. One is a search algorithm, another is a scoring function. So in the first docking, we have to do the search algorithm. So in the search algorithm, it generates the multiple poses and conformations, positions and orientation of the ligand within the active site, and see how many number of conformations and the orientation it is going to interact in the pocket, it will search. There is another mode is called as a scoring function. The scoring function is to identify the most likely pose for the individual ligand. So one ligand has got multiple poses, another ligand has got very few pages. That is the reason. So the most likely pose for the individual ligands, we need to assign the score. So first we are generating the poses, then we are assigning the score. So to assign the priority order, to set the diverse ligand locked in the same position, estimate the binding affinity. When the score is very high, the drug-like moiety is very high. So we will pick up first for the further progressing it. So previously what we have done, the filter mechanism, here we will further filter them in the docking studies, reduce the number before going to our regular lab, that is animal studies and clinical trials. In this particular journey, the search space is very high. What we are doing in the journey, many are there. So the difficulty with the protein ligand tagging is a part due to the fact that it involves the many degrees of the freedom in the 360 dimensions. So the translation and rotation of one molecule related to the another, so involves the 6 degrees of the freedom. So these are in addition to the conformational degrees and freedom of both ligand and the protein, both are flexible in nature. They will be having a different conformation, makes the complexity better, and it is increasing the challenges. So the solvent may also present like water effects, which will be present significantly role in the protein ligand geometries. So many cases we are having a polar surface area. Obviously, the water will influence the water effect. We cannot ignore there. So the search algorithm generates poses, orientation of a particular conformations of the molecule in the binding site. It tries to cover the search space, if not exhaustively, then extensively as possible, because we may miss sometimes the certain poses also, because we cannot make multiple freedom, that is multiple degree spaces, very less narrowing it, it will increase the time. That is the reason it may be having such possibility to missing a positive out also sometimes. So there is a trade-off between the time and the search space coverage. That is the reason we need to select a space between how many conformation we want to take it. So, and what is the degrees? So we want to give in a space between the one conformation to another precision. What is the degrees we want to have it, so that we can compromisely have the less time, also more coverage also. So what are the DALK algorithms which are present and which are being used for this particular function is, so it is based on the shape complementary for the rigid shape complementary and the rigid ligands. So in the another algorithms which are present is based on the fragment based methods, where we have got a dark and flex. Monte Carlo simulation annealing, it will be projecting near positives, and it will suggest you the modifications in the structure that will give you as a drug also in the Monte Carlo simulations. So for that purpose, we have got autodock and affinity and ligand fit, QSP flow also present. So genetic algorithms based on completely starting from the scratch and making it how it is each the journey is bringing into the drug, where we have got a gold and autodock, and a systematic search, where step manner we are going to incrementally increasing the activity we can do by the Fred, that is OpenAI, and Glide, that is Schrodinger software. So these, when we are having a systematic and genetic algorithms, when the complexity is more, the cost will also be very high. That we need to see. So ultimately, by using these softwares, we are having a dock. You see this particular picture. The chemical one is represented in that three-dimensional array in this particular right side of your slide. If you see it, how this is having a three-dimensional as a circles. So here each atom previously in the chemical space, we are representing with the Euclidean distances. So now we are converting into the circles, spheres with varying diameters. So that is the affinity scores. Based on this, now we are going to see. In the two-dimensional, we are finding with the Euclidean spaces and triangles and the edges. Now we are seeing the radius of the sphere, which is present with the affinity of each atom, which is present in the dock. So the rigid docking is based on the shape. A negative image of the cavity is constructed by filling it with the spheres, and spheres of varying size depending upon the atom type. Each touches the surface of at 2 points, and every back, front, top, everywhere it is having going to have a interactions, and the sphere overlaps will also be present when you see from the top. So the center of the sphere becomes the potential location for the ligand. Obviously, that is going to govern the distance, Euclidean distances, previously what we have seen in the chemical search methods. So like this, we are going to have the rigid docking type. So that we are going to have number of conformations are going to be less to fit this ligand into the receptor. So after that, what we are going to do, we are going to similar moieties. If you see from the top to bottom, all the shapes are appearing the same with three-dimensional space, but the chemical similarity not present. They are completely different, but they are occupying in the rigid manner in the similar spherical shapes. So that is the reason we have got a pharmacophore mapping now. We are going to find a new leads with this particular concept. So the ligand atoms are matched to the sphere centers, so that the distance between the atom equal to the distance between the sphere centers. So we have got a linkages of our chemical search with pharmacophore mapping now. So the methods are used for the position of the ligand within the active site. If there are no steric classes, the ligand is scored better. And many different mappings poses are possible to see the fit and assign the scores. Each score is scored based on the goodness of the fit, because it is having irregular shape. So goodness of fit will be present when it is occupying the pocket completely, and when it is rotating, and it is also not having a problem for the free movement and interactions are still present. Highest scoring pose is represented to the user. That will be assigned and give to the user as a output. So we are going to get the final of our journey, where we are going to have the energy minimizations, and our journey is going to end now. The chemical is going to converting into a drug after seeing this one successful. So the energetics of the protein ligand binding, we need to see the free energy of total binding, free energy of the Gibbs free energy of the total binding, drug receptor binding. So there are electrostatics, that is the hydrobonding interactions, van der Waals forces, they are called as dispersion forces, and hydrophobic interactions, they will be like dipole-dipole interactions, desolvations, surfaces are buried within the protein, and the ligand have to be desolvated, removing the water effects. Conformational changes to protein and the ligand by the multiple rotations based on the probatum. Ligand must be probably that is properly oriented and translated to interact to form a complex. So this is all the things, and we are going to see the loss of entropy of the ligand due to the fixing in the one conformation. We are going to study the energy of the binding. So the energy of the total energy of the binding, that is change in the free energy, is contributed by the solvent effect, conformational changes, intrinsic factors, rotational bonds, and vibrational one. They are going to have the impact onto the total binding. So some are having the negative, some are having the positive. Ultimately, together we are going to get one score. That score is going to give us the whether the Gibbs free energy is very high or low. The high scores means which is having less stability. We have to drop the moiety there itself. The low scores which are going to represent the one which is going to have a better affinity. So in conclusion, Autodoc, Doc, E-hits, Flex, Fred, Glide, Gold, Ligand Fit, XP, UQXP, and Surflex Doc, among the others, all are getting used for our molecular docking. Different scoring functions, we are going to have a different search algorithms, and different approaches are based on the what software we are using, what type of approach we are assigning. Based on that, we are going to have a many softwares. I have already given you one assignment. You will get many more software you are going to submit on Monday. So that will give you idea that many more software also present in addition to the what we have got in this particular presentation. So wide range of the virtual screening techniques are developed, but the performance of the different methods varying with the different data sets. So the increased complexity in the descriptors and method does not necessarily lead to the greater success. That is the reason we have to use a combined effort. That is different approaches can lead to a improved results instead of relying on only one, only chemical based or the only not varying the energy minimization, such things which will be resulting into a false positives. So computational filters should be applied to remove the undesirable compound in the early stages, so that we will be having workload less. By this, we can result into a drug with a guarantee, and we are going to have a success rates very high in the animal studies further and the clinical studies in the human beings, and resulting into a more generation of the revenues in the less point of time by using the computer-aided, computer-assisted drug design. So these are all many references. So with this, our journey of drug design is coming to